In today's class, we are going to study about classical experiments which were done late back to establish DNA as the genetic material. Now, how the scientists determined that it is actually the DNA out of all the biomolecules that are present in the cell which acts as a genetic material. सेल के अंदर प्रोटीन्स हैं, कार्बोहाइड्रेट्स हैं, फैट्स हैं, न्यूक्लिक एसिड्स हैं, बहुत इनऑर्गेनिक मटेरियल है. So what is that which is actually the hereditary material, and how it was finally established that DNA is the genetic material? So there were many groundbreaking experiments done by Griffith, by Avery, his friends McLeod and McCarthy, Hershey and Chase, which went against the notion that proteins were genetic material, and they established the DNA as genetic material. Till then, people were mostly thinking that proteins were the part that actually exist as the genetic material, but their experiments established the fact that it is the DNA which is the genetic material. First of all, we will discuss the Griffiths transformation experiment. Now, in 1928, Griffith conducted experiments in which he used bacteria Streptococcus pneumoniae. This bacteria causes pneumonia disease, and he also used mice. Now, Griffith, while he was conducting experiment with Streptococcus pneumoniae and mice, he was actually trying to develop a vaccine against this bacteria. But during this experiment, he found some interesting results which led to the establishment of the fact that DNA is the genetic material within the cell. Now what he did, he used two strains of DNA. One is called as the R strain and another one is the S strain. R strain ये दोनों स्ट्रेप्टोकोकस न्यूमोनिये ही है, लेकिन R strain वाला स्ट्रेप्टोकोकस न्यूमोनिये it does not causes disease, and smooth strain it cause disease, the pneumonia. And because when grown on medium, these R strains, their colonies appear to be rough. Therefore, this is called as the R strain, and the S strain, its colony appear to be smooth because of the presence of a polysaccharide coat around it. And this is actually, S strain is actually the bacteria which causes the pneumonia. Now, this is the experiment conducted by Griffith. What Griffith did is he took living R cells, the diseases, the non-virulent strain of the bacteria. Now, this living R cells, they were injected into a healthy mouse. And finally, what it was seen that the mouse did not die, and he could isolate the R cells from the body of the mouse. They were replicated in the body of the mouse, but they did not cause the death of the mouse. Then he also he also injected the living smooth strain which caused disease, which was known to cause disease, into mice, and the mouse died. And from the mouse he could isolate the smooth cells. Means this bacteria, when it was injected into the mouse, it grew in number, and from here from the dead mouse, this living cells of the S strain were taken out. They were found in the dead mouse. Now what he did, he killed these living S cells. Now because he heated that because of the heating, these cells were dead. When he injected these dead S cells into a healthy mouse, then the mouse did not die, and nothing was received from the no S cells were found in this mouse, because they were dead, they did not replicate, so they were not found in its body. But when these dead cells, they were mixed with the living R cells together and then injected into mice. Now you see, the R cells do not cause disease. The killed smooth S cells do not cause disease. We saw in this case and in this case. But when both of them were mixed together, then the mouse in which it was injected, that mouse died. And from this mouse, to a great surprise, he found that he got some S cells and some R cells. अब R cells तो इसमें इंजेक्ट किए थे तो R cells मिल सकते थे, but from where did these S cells come from? That was the point. So from this experiment, Griffith concluded that there is something in these S cells, dead S cells, that could enter the R cells, and the R cells were then transformed to S cells. This material which was causing the transformation of the R-type strain, non-virulent strain, to S-type virulent strain, that was called as transforming principle by Griffith. And this effect was that time called as Griffith's effect, but later on it is, till today we famously know this phenomenon as transformation, in which the material from one bacteria that enters into another bacteria, and that is the genetic material. So now, because since till this experiment it was not known what is the nature of this material which is transforming the R-type bacteria, because the R-type changed to S-type, that means it got some information, then only it changed to S-type, and that information is the genetic material that was hypothesized by Griffith only, that what this transforming principle is actually the genetic material. The transforming principle is actually the genetic material what Griffith proposed. Now, after so many years, when every MacLeod and McCarthy, they conducted one another experiment which was useful in telling that what part of the cell is actually the transforming principle or the genetic material. For this, they did a famous experiment which we know by every MacLeod and McCarthy's experiment in bacteriophage experiment. In this, every McCarthy and MacLeod's experiment, not the bacteriophage, that is the next Hershey and Chase, but in every McCarthy and MacLeod's experiment, what they did is they took heat killed S strain. S strain जो है we know that causes disease, लेकिन it was heat killed, and the capsule components of this particular bacteria they were removed. इसमें से lipids और carbohydrates जो है वो remove कर दिए. बाकी ये जो अब solution है जिसमें heat killed ये बैक्टीरिया है, उस बैक्टीरिया का प्रोटीन, आरएनए और डीएनए वो सब इसके अंदर, इस सोल्यूशन के अंदर प्रेजेंट है. नाउ दिस सोल्यूशन वाज डिवाइडेड इनटू 3 पार्ट्स. वन ऑफ द पार्ट वाज केप्ट एज सच, जिसको कोई ट्रीटमेंट नहीं दिया, लेकिन वन ऑफ द पार्ट इन दैट प्रोटीएज वाज एडेड. Protease is the enzyme which will degrade the proteins. So जहां पे प्रोटीएज डाल दिया उस सैंपल में अब प्रोटीन्स नहीं बचे हैं. तो नेक्स्ट वाला जो है हियर, इन दिस they added ribonuclease. Ribonuclease will break the ribonucleic acid, RNA. So in this sample, RNA was not found. Here in the last sample, they added DNase, deoxyribonuclease, which degraded the DNA. So in this sample, DNA was not present. So only in this sample, DNA was not present and DNA was present in all the other three solutions. Now what they did is, next they added this this particular treated sample, ये जो these all four samples, now they were used to they were added to the R cells. R cells के culture में these solutions were added separately. जहां पे R cells में ये untreated solution डाला, वहां पे R cells जो है they were transformed to S cells. Okay, so transformation occurred in this case. That means something from this solution could transform R cells to S cells. So here transformation occurred. Now in this sample, what happened? This solution was added to R cells, and the R cells, they were again transformed to S cells. That means something was here that could transform these R cells into S cells. And it was also concluded that proteins are not required to transform the R cells to S cells, क्योंकि यहाँ पे प्रोटीन्स प्रेजेंट नहीं है, इसमें प्रोटीन्स है. ठीक है, इसमें डीएनए और आरएनए है. डीएनए और आरएनए में से ही कोई चीज है जो इसको आर सेल्स में कन्वर्ट कर रही है, प्रोटीन्स नहीं कर रहे इसका मतलब. लेकिन जब बिना आरएनए वाला सैंपल, जिसमें आरएनए भी नहीं है, जब उसको यूज किया आर सेल्स के साथ में, तो इन दैट केस ऑल्सो द एस सेल्स they were received. The R cells could be transformed to S cells. That means that not even the RNA and not even the protein that is needed for transformation of R cells. Now we have to see whether the DNA was there which could transform or not. Now, when the sample in which there was no DNA, that was used that was added to R cells, in that case S cells were not formed. That means जहां डीएनए नहीं है, वहां आर सेल से एस सेल नहीं बन सकता. आर सेल को इंफॉर्मेशन नहीं मिल सकती एस सेल्स में ट्रांसफॉर्म होने की. सो दिस established that DNA is the material which will transform the R cells to S cells. Or we can say that DNA is the genetic material. There was still another experiment by Hershey and Chase, which is called as Hershey and Chase bacteriophage experiment, which was again a support to the fact that DNA is the genetic material and it's not the protein which is the genetic material. So this experiment, Hershey and Chase used bacteriophage. T2 bacteriophage use किया. This is the structure of the bacteriophage, two-dimensional and three-dimensional. It has a icosahedral head, then it has a tail, a collar and spikes to infect the bacterial cell. Now in this bacteriophage, there is presence of a protein coat and DNA. And how this phage infects the bacterial cell? It attaches on the bacterial cell, then it allows something to pass from its body into the bacterial cell. This something which passes into the bacterial cell, today we know that it is DNA. But at that time, during the time of Hershey and Chase, it was not known what material is that which goes inside to the cell, क्योंकि जो मटेरियल अंदर बैक्टीरिया के अंदर जाता है it carries the information to make new phage particles, और ये मटेरियल बैक्टीरिया के डीएनए में इंटीग्रेट होके बैक्टीरिया की पूरी मशीनरी को यूज करता है और छोटे छोटे और नए बैक्टीरियोफाजेस बनाता है। ये बैक्टीरियोफाजेस फाइनली बैक्टीरियल सेल को लाइज करके जो है दे आर then released from the bacterial cell. But today, as I told you, we know that it is DNA which enters the bacterial cell. But when Hershey and Chase were conducting this experiment, it was not known whether it is the DNA that goes inside the bacterial cell or whether it is the protein. लेकिन जो चीज अंदर जाती है दैट इज ऑब्वियसली द जेनेटिक मटेरियल ऑफ द वायरस, क्योंकि अगर वो जेनेटिक मटेरियल नहीं होगा तो नए वायरस पार्टिकल बनाने की इंफॉर्मेशन उसके पास नहीं होगी। सो इट वाज नोन दैट इट passes its genetic material into the bacterial cell, but whether it is DNA or protein, that was not known. So in their very famous experiment, you can pause it and read it. I will explain from the figure. What happened in their famous experiment, Hershey and Chase, he grew one sample of virus on the radioactive sulfur, जो S32 से जो है it was labeled S35. The radioactive isotope S35 was used to label one of the sample of bacteriophages, and as we see sulfur जो है वो हमेशा वो प्रोटीन्स के अंदर प्रेजेंट होता है और डीएनए में प्रेजेंट नहीं होता है। तो अगर ये वायरस का एक जनरेशन S35 कंटेनिंग मीडिया पे वायरस को ग्रो करेंगे, a media which contains the isotope S35, then the bacteriophages which will be received in this particular culture, they all will have the sulfur labeled as S35, और जहां जहां भी ये मैक्रोमोलिक्यूल्स में सल्फर होगा वो S35 होगा। तो प्रोटीन्स में सल्फर होता है तो प्रोटीन्स में भी जो है S35 की रेडियो एक्टिविटी आएगी। डीएनए में नहीं होता है तो डीएनए जो है वो जो रेडियो लेबल्ड नहीं होगा। लेकिन अगर ये P32 कंटेनिंग आइसोटोप जो है, अगर कोई ऐसा मीडियम है जिसमें P32 जो है फॉस्फोरस पूरा P32 वाला डाला है, रेडियो एक्टिव फॉस्फोरस डाला है, इन दैट केस द DNA of the bacteriophage, it will change to radioactive because it will contain the phosphorus which is radioactive, the P32. लेकिन phosphorus is not present in the proteins, so proteins will not be labeled. Now, as we know, during the infection process, there will be adhesion of this bacteriophage on the bacterial cell and it will pass its genetic material inside. जब ये जेनेटिक मटेरियल इसको इन्फेक्शन अलाउ किया बैक्टीरियोफाज को, देन बाय ब्लेंडिंग, सिंपल मिक्सी जो होती है उसमें ब्लेंड करके व्हाट दे डिड इज, ये जो बाहर का मटेरियल है जो बाहर रह गया है वायरस का, उसको बैक्टीरिया के सेल से अलग कर लिया। अब क्या बचा है, वो मटेरियल वायरस का जो बैक्टीरिया के बाहर रह गया है और बैक्टीरियल सेल प्लस वायरस इसका जेनेटिक मटेरियल रह गया है। जब सेंट्रीफ्यूगेशन किया तो बिकॉज दिस वाज ए लाइटर बैक्टीरिया, जो वायरसेस के घोस्ट रह गए हैं बाहर वो जो है क्या है, लाइटर है, तो दे वर सेपरेटेड फ्रॉम द रेस्ट ऑफ द बैक्टीरियल सेल्स। लेकिन बैक्टीरियल सेल्स के अंदर जेनेटिक मटेरियल भी है वायरस का। नाउ व्हाट वाज सीन दैट इन द सैंपल where the sulfur was radio labeled, in that case there was no sulfur present in the bacterial cells, but जो घोस्ट वाला पोर्शन था सेंट्रीफ्यूगेशन से जो अलग किया उसमें सल्फर डिटेक्ट हुआ, and in the other sample in which the infection was done using phosphorus labeled bacteriophages. In that case बैक्टीरिया जो मिले उनमें phosphorus का radio labeled phosphorus mila, but there was no radioactivity in the ghost. That means जो अंदर गया बैक्टीरियल सेल्स में वो था डीएनए, तभी तो रेडियो एक्टिविटी जो है वो बैक्टीरियल सेल में इस वाले केस में मिली और इसमें नहीं मिली. So this experiment also helped in establishing that DNA it acts as the genetic material, and by today we all are very much convinced, all science in today's day, it is based on the acceptance that DNA is the genetic material, because there are now millions of experimental proofs, even microscopy has been done. So all that we know today is established science, जो कि ये छोटे छोटे एक्सपेरिमेंट्स जो हुए थे, इन एक्सपेरिमेंट से आगे चल के जो है ये और ज्यादा जो है इट इज नाउ established.